Welcome to Lecture 20, where we will go over the structure and function and replication of DNA. Thomas Hunt Morgan was an important geneticist looking at the structure of DNA. He discovered that genes were located on chromosomes. However, at the time it was unknown what chromosomes were made of. Many scientists thought that the DNA molecule was actually protein-based. Frederick Griffith followed up with experiments where he looked at Streptococcus pneumoniae and mice. He identified that bacteria can be transformed by acquiring genetic material from their environment. This genetic material from their environment would eventually be incorporated into their own genome, transcribed, and translated. This slide here outlines Griffith's experiments. In his experiment, he used two different strains of Streptococcus pneumoniae bacteria, S and R strains. The S strains of the bacteria were considered to be pathogenic, releasing a toxin that would lead to death, whereas the R cells were considered to be non-pathogenic. In his experiment, he had three different control groups. The first control group consisted of living S cells. He took the living S bacteria cells, injected them into the mice, and because this bacteria strain was considered pathogenic, the mice ended up dying. The second control group was considered a non-pathogenic control. In this group, he injected the mice with living R cells. Because the R strain is non-pathogenic, the mice ended up being healthy. In the third control group, Griffith injected mice with heat-killed S cells. In this group, the mice ended up healthy. This was also considered to be a non-pathogenic control. Because the S cells were heat-killed, they were not transcribing or translating their genetic information. Finally, in his treatment group, he took a mixture of heat-killed S cells, mixed them with living R cells, and injected this mixture into the mice. In this treatment group, the mice ended up dying. Pathologically, the mice ended up having living S cells circulating in the system. This supported the idea of bacterial transformation, such that the living R cells were able to uptake the genetic information from the heat-killed S cells, and thus were transformed into living S cells. The previously outlined experiments were important in demonstrating that bacteria can undergo transformation. However, it was currently unknown whether that transforming agent was DNA or protein-based. Avery and McLeod, important scientists, discovered that the transforming agent was indeed DNA-based. Hershey and Chase did a series of important experiments that also investigated this process. However, they used bacteriophages, which consists of a virus that infects bacterial populations. They grew the virus in two different conditions, in a medium with radioactively labeled sulfur or in a medium with radioactively labeled phosphorus. They used these two mediums because sulfur ends up getting incorporated into proteins. Remember, disulfide bridges is a very important thing to form for a protein to take on its final conformation. Phosphorus, on the other hand, is a main component of the DNA backbone. As a result, phosphorus gets incorporated into the DNA. These two properties allowed these scientists to determine what the transforming agent was and how it infected bacterial cells. Their main question was, when a phage infects a bacteria, what enters into the host cell? Is it protein or is it DNA? This slide gives an overview of Hershey and Chase's experiments. Note that in the top left corner of this slide, we have the structure of a bacteriophage. Note that the DNA is surrounded in the phage head, and this is then attached to a bacterial cell, where the transforming agent will be injected into the host bacterial cell. Within his experiment, he had two different batches: bacteriophages that were raised in radioactive sulfur and bacteriophages that were raised in a medium with radioactive phosphorus. Note that if the medium contained radioactively labeled sulfur, this would result in the phage proteins being radioactive, And if the bacteriophage was grown in the phosphorus batch, this would mean that the phage DNA was in fact the labeled portion. He then allowed these phages to infect bacterial cells. He then agitated the medium in order to separate out the phage parts from the cells. He then put these samples into a centrifuge in order to separate out the sample. In the centrifuge sample, there would be a pellet at the bottom that contained the heavier elements, which would be consistent with the cells. The other portion would be considered the supernatant. The scientists then measured the radioactivity in both the pellet and the liquid to try and determine what the transforming agent was. They determined that the phage radioactively labeled proteins were found in the supernatant or outside of the cell, whereas the phage labeled DNA was determined to be in the pellet, which consisted of the host cells. This allowed them to determine that the transforming agent was, in essence, DNA-based. Erwin Chargaff was another important scientist that was studying the structure of the DNA molecule. He measured the components of DNA across many different species, specifically looking at the proportion of DNA that was attributed to each nucleotide base. He determined that each species has different amounts of A, T, C, and G. This was important in disproving the common theory that at the time was prevalent. It was originally believed that DNA was simply long repeats of a similar sequence, and that the complexity of an organism had to do with the fact that more DNA was present. He demonstrated for the first time that the DNA itself differed between species. He also noticed an important finding that the percentage of adenine equaled the percentage of thymine, and similarly the percentage of guanine equaled the percentage of cytosine. This is now known as Chargaff's law, such that the bases pair up in a consistent way. This provided very important information about how these bases were paired up in a DNA molecule. There were many important scientists also studying the structure of the DNA. This included James Watson, Francis Crick, Maurice Wilkins, as well as Rosalind Franklin, and all of their findings were crucial in determining what the structure of DNA actually was. The first correct model, however, was proposed by Watson and Crick. They determined that DNA existed as a double helix, such that the two strands of DNA run anti-parallel to each other. Remember, one strand will run from 3 to 5 prime, while the other one will run from 5 to 3. They also determined that the sugar and phosphates face outward towards the molecule, interacting with the surrounding water. However, the nitrogen bases would face inward, and they in fact form hydrogen bonds with each other to pair up according to the base pair rule. These bases pair up such that adenine binds with thymine with 2 hydrogen bonds, whereas cytosine and guanine form 3 hydrogen bonds. This image here depicts Rosalind Franklin and her important work on understanding the structure of DNA. She was a chemist studying using X-ray diffraction techniques to visualize the DNA molecule. We can see from the photo to the right, this would be equivalent to looking down a DNA molecule. We can see the presence of 2 phosphate backbones, one on the top and bottom of the image. We can also see the nitrogenous bases pairing up within the middle of the molecule. Her work was pivotal in allowing for Watson and Crick to determine the actual structure of a DNA molecule. Their original theory at the time was that the DNA consisted of 3 different strands, and the phosphate backbone would be on the interior of the molecule, whereas the nitrogenous bases faced outward. After seeing Rosalind's photo 51 and after obtaining some measurements regarding the spaces between the different nitrogenous bases and phosphate backbones, they were able to determine that DNA had 2 phosphate backbones such that the nitrogenous bases faced inward to allow for the formation of a double helix. Their physical model that they built for a DNA molecule was pivotal in understanding what the structure actually was. They had subsequently received a Nobel Prize for their findings for the structure of the DNA molecule. We're now going to take a closer examination of the structure of a DNA molecule. Note that we have the sugar phosphate groups acting as a backbone for the molecule. Also note that we have two ends designated for the strand, 5' and 3'. Remember, as discussed in previous lectures, 5 versus 3 refers to what carbon is exposed at the end of the chain. The 5' carbon would be exposed at the 5' end, and the 3' carbon of the sugar would be exposed at the 3' end. Also note that there are 4 different types of nitrogenous bases: thymine, guanine, cytosine, and adenine. And these bases will then pair up with the corresponding base according to Chargaff's law. We see in the structure of the DNA double helix that we have anti-parallel ends. When one strand runs 5 to 3, the other strand will run 3 to 5. Also note that we have the base pair rule holding true, such that adenine binds with thymine and cytosine binds with guanine. This will occur throughout the double-stranded DNA. We also know that the two strands of DNA are not considered to be identical to each other. Rather, they are complementary to each other. They run anti-parallel in opposite directions, as stated previously, and when one strand has A, the other strand would have a T, according to Chargaff's law. This structure makes easy copying according to what is termed the semi-conservative model. The semi-conservative model of DNA replication states that the two strands of DNA will unzip, separate, and both will serve as copy templates to create a new strand of DNA. Originally, there were 3 possible models of DNA replication that were proposed: conservative, semi-conservative, as well as dispersive model. Meselson and Stahl were important scientists that tested out the validity of these 3 models. In order to test these models, they used radioactively labeled nitrogen in order to label the DNA. They were able to prove that the semi-conservative model was indeed true. Let's take a look at their experiments. These are their hypotheses based on the 3 different models of DNA replication. They took a parent cell, labeled the DNA in that parent cell, and then traced where these radioactively labeled strands were present after 2 successive rounds of division. During the first replication, we can see the difference between the 3 models. In the conservative model, after the first round of replication, the parent DNA would only be found in one of the cells. The conservative model would state that both DNA strands would act as a template, however, they would remain together. As a result, only one of the daughter cells would contain the radioactively labeled DNA, whereas the other daughter cell, after the first round of division, would contain entirely new DNA molecules. In this model, after the second round of replication, we would see the similar pattern holding true, such that when the radioactively labeled daughter cell would undergo the second round of division, if the conservative model were to hold true, then only one of the daughter cells from the second round of division would have the radioactively labeled DNA, whereas the remaining daughter cells from the second round would all have new DNA molecules. Let us now go to the bottom of this diagram to look at the dispersive model. The dispersive model would state that the parent strand, fragments of the parent strand, would be used as templates for new DNA molecules. As a result, if this model were to hold true after the first round of replication, we would note that both daughter cells would have both parent DNA as well as new DNA. Subsequently, after the second round of replication, each of the daughter cells produced in the second round of replication would have both parent DNA that was radioactively labeled as well as new DNA. Now let's discuss the semi-conservative model. Remember, in the semi-conservative model, it states that the DNA molecule and strands would be unzipped and each of these strands would act as a template. As a result, after the first round of replication, each of the daughter cells would contain a parent strand that was radioactively labeled in conjunction with a new strand. Similarly, if these cells were to undergo a second round of replication, we would see that in both populations from both of those original daughter cells, they would divide in such a manner that one of their daughter cells would have the parent strand as well as a corresponding new strand, whereas the other resultant daughter cell from the second round of replication would have entirely new DNA strands. After conducting their experiment, they found that the pattern depicted in the semi-conservative model did indeed hold true for both the first and second round of replication. We're now going to take a look at the mechanics of this process. Keep in mind that the overall genome difference between prokaryotic and eukaryotic cells. A recurrent theme in this class is being able to compare the differences and similarities between prokaryotic and eukaryotic cells. When discussing DNA replication, most of these differences come down to the state of the genome. Remember that E. coli or prokaryotic cells have one circular chromosome, while humans have many linear chromosomes. As a result, this causes a distinct difference in the DNA replication process, such that prokaryotic cells will only have one origin of replication, while eukaryotic cells will have thousands. This is because they have more complicated DNA and they also have linear chromosomes, which allow for multiple origins to be present. An origin of replication is simply a region on the DNA sequence that can be recognized by the proteins that begin the process of DNA replication. At the location of the origin, the strands will be melted, meaning that the hydrogen bonds will be dissolved and the strands will be pulled apart. When these strands are pulled apart, this will lead to the formation of a replication bubble. Note that the replication process occurs in both directions. We say that this process is bidirectional. This means that we will have at each end of the replication bubble the presence of a replication fork, and DNA replication will occur in both directions at each fork. This diagram here depicts the difference between the origins of replication in prokaryotic versus eukaryotic cells. In prokaryotic cells, the image on the left, we can see the formation of a replication bubble at the origin of replication. Once the strands begin to be pulled apart, the enzymes responsible for DNA replication can access the DNA. Also note that we have bidirectional replication occurring, meaning that at both replication fork we will get DNA replication occurring. Once the replication bubble increases, more DNA can be accessed, which means that more DNA replication will occur. We can see the growth of the new strands, which are located in light blue, and we can see that both of the parent strands, located in dark blue, are being used as a template for the new strands. Once the DNA is copied, the genome can separate such that each of the daughter cells will have an original parent strand as well as a new parent strand. If we take a look at this process in eukaryotic cells depicted on the right, we can see that there are multiple origins of replication within one linear chromosome. We can also see that the overall process is similar, meaning that a replication bubble will form, replication forks will also form, and the DNA replication will occur bidirectionally. The bubbles will increase and the DNA strands will be replicated until they eventually meet with each other. These fragments will then be glued to ensure that we have a continuous copy of DNA. Also note that we get the similar result in the DNA of the daughter cells, such that each daughter cell will contain an original parent strand that will be held in close association with the new DNA strand.